we finished up talking about the difference between metabolism and anabolism. So today I'm going to be talking about the nutrients. And the nutrients are the first three here. I'll explain more about that in a minute. But the best way to conceptualize nutrients is, they're these big chains, what are called polymers. And they're made up of individual links we call monomers. And you just gotta get used to this. Well, when we're talking about catabolism, we're talking about breaking things down from things that are larger to things that are smaller. And when you do that, energy is freed up. And then this is actually going to be the topic. I'm going to have this on the whiteboard several times. Okay? Because it's really key. And then anabolism is building up. So you take the links and you build the bigger chains. Does that make sense? So here are the 4 biologically important macromolecules on this slide. There they are listed there. Now the two main topics for lecture exam 1 is how your cells will take these links and make ATP energy out of them. And there are only 2 types of nucleic acids. There's DNA and there's RNA, and we already saw that ATP is an RNA nucleotide. But how our cells use these guys to make ATP is one big topic, and then the other big topic is DNA. So a lot of cell chemistry on lecture exam 1, which is you just have to do. That's how physiology is. And then we finished up talking about mechanical work. Okay? Are we ready to go? All right. Let's talk about the chemistry of the cells. Protoplasm is just the basic stuff in the cell. It can either be inside the nucleus, that's nucleoplasm, or inside the cell, but outside the nucleus, that's cytoplasm. This is A plus B equals C. Cytoplasm plus nucleoplasm equals protoplasm. This one would be today. Yeah. Is Mario your friend?
Yeah.
Okay. Yeah, you need to come earlier. Yeah, and you don't get the ad codes till kind of start making them late.
Equals protoplasm?
I'm gonna go then, because I won't be able to stay.
Okay, then you can have it. All right.
Sorry.
Everybody got that? A plus B equals C?
Mm-hmm.
Okay, the 5 most common chemicals in the cell. First is water. Now, like we said, your body is 60% water by weight. Then you have ions, or electrolytes, same thing, and actually throughout. Ions. Well, the reason why ions are hydrophilic is because water is polar. So if you've got a negative ion, then the little positive side of the water molecules will point toward it. If you are positive ion, then the negative acid of the water molecule will be up against it. And then, this is the reason why these are the 3 nutrients. Because you have so much of those in your cells. Okay? That's why you've got to eat a lot of that. Now, I'm going to just give you a little bit of brief information here, and then we've got to get going to the PowerPoint. If you understand how our bodies work, like our digestive systems, when you eat these large chains. For example, A lot of people believe you can take melatonin to help you sleep. This is why you guys need to get educated. Because we have a whole generation of people, you might be one of them, who does not know the difference between true science, pseudoscience, non-science, bad science, or evil science. That's why some people believe vaccines cause autism. That was evil science. The scientists actually doctored his results to show that. You guys know GNC store? 85% of the products in GNC are there because of placebo effect. That whole chain is making money hand over fist on things that they actually do not have evidence that it works in double-blind studies. How do they succeed? Because we have a whole generation of Americans who can't tell the difference between true science and pseudoscience. When you eat a protein, your digestive system are going to reduce it to single links, and only single links can be absorbed. If you take melatonin, that's a specific protein that never sees your blood. The only thing that goes into your blood are the amino acids from melatonin, which have no effect on your sleep. If you sleep better because of melatonin, it's placebo effect. Now, usually with patients, I will never take their placebo effect away. They tell me, oh, this thing really works. I say, that's great, I'm glad you found something that works for you. You should recommend it for your other patients. You bet. Hell no, I won't. That'd be malpractice. And I'm not gonna take their placebo effect, so if you actually take melatonin for sleep, I'm sorry, I just took it away. Watch what happens now. It's not gonna help you. Placebo effect is real. Documented, double-blind, actual effects, how things work. But on this case, you do need to eat those 3 macromolecules so that your digestive system will reduce it down to single links, and now it'll go into your blood and your cells can build up their own chains. Everybody got it? You eat these, it gets broken down, these get absorbed, then your cells will build these guys back up. Everybody got how that works? Okay. And then it builds them up so much that it is in the top 5. All right, we do have to talk about carbon. I mentioned carbon last week when I was talking about methane and how it produces things that are non-polar. Let me just give you a little heads up in case you do have to take organic chemistry. Organic chemistry is the chemistry of carbon. It has nothing to do with pesticides or labels you see in the grocery store. Everybody got it? That's not what organic means. Organic means carbon. So when I was showing you ATP, and when you split off that last phosphate group, I said that's ADP, and inorganic phosphate. Do you guys remember that? It's inorganic, not because it has pesticides on it. It's inorganic because it has no carbon. Everybody got it? All right, now because carbon has 4 electrons in the outer shell, it's right in between 0 and 8. And because of this, carbon will form 4 covalent bonds. That's the sharing, electron sharing like with water, H2O. But the thing about these 4 bonds is they try to get away from each other as far as they can in 3 dimensions. And it's because of that, carbon's covalent bonds are very stable because they are symmetrical in 3 dimensions. Extremely stable covalent bonds. Carbon also has one of the lowest activation energies of all the atoms. What does that mean? That means it'll bind with anything, including itself. Out of 20 million known compounds, over 85% are organic. That's why organic chemistry is a big topic. Organic chemistry is not about the chemistry of cells. The chemistry of cells, that class is called biochemistry. Organic chemistry is the chemistry of carbon, whether or not it has to do with cells. Most things do, but not everything. It's only about carbon. Are we clear? Because of these bullets. Because it has got the lowest activation energy of all atoms, it binds to anything. It's extremely versatile. Or for that third bullet, you can say carbon is a whore. It'll hook up with anything. Because of that, that atom is the backbone of these molecules. You will see carbon throughout. And if you are a Trekkie, don't raise your hand because people laugh at you. But anything in this column should be able to do the same thing, which is why in science fiction sometimes people write about silicone-based life forms because it's in that same column. Everybody got it? Okay. In fact, in organic chemistry, they get so sick of writing C, since it's the atom of that's the backbone for all these frames. They don't even write the C. Every place you see a corner is a carbon. They just say, I'm not gonna write all the C's out. And they don't. So if there's other atoms like nitrogen, they have to put the N there, but where there isn't any letter, that's a C. Do you see how it is the backbone of all these major molecules? Any questions? All right, let's talk about enzymes. Enzymes are proteins, so all enzymes actually go here. Now, all enzymes are proteins, not all proteins are enzymes. Everybody got that? But it functions as what's called a biological catalyst. So when we're talking about cell metabolism, where we're gonna explain how your cells turn this into molecules of ATPs, we're gonna have a whole sequence of chemical reactions. Those chemical reactions are not random. They are driven by these specific enzymes. Okay, so that's why I gotta cover some of this. Everybody, look at me. Today's lecture is setting up Monday's lecture, where I'm going to actually talk about metabolism. So I'm covering a bunch of preliminary background, okay, today. So pay attention. If you don't get this, it's going to be harder to understand when I get into metabolism. So this is important. These reactions are not random. They are specifically driven by enzymes. A catalyst is something that speeds up reaction rates, chemical reactions rates, without being permanently altered or used up. It says part of it here. Speeds up chemical reactions without being used up in the reaction. It is not permanently altered. In the case of enzymes, they are temporarily altered. But when the chemical reaction is done, it snaps back in its original shape. So it's not permanently altered. Everybody got it? Only temporarily changed. What all catalysts do, and this is what enzymes do, is they lower the activation energy of chemical reactions. Do you guys need for me to explain that?
Yes,
Yes.
please.
Okay. This is going to be a graph of potential energy. Remember when I talked about secondary active transport? And I drew that picture of the semi with sodium going up to the top of the hill? It had to burn gas to get there. Once it's there, elevated, it now has potential energy. You can turn the engine off and have it coast downhill, and it can take a pickup up the hill, okay? It has energy once it's up there. This graph is very similar to that. Things that are up in here have high potential energy. Things that are down here are low potential energies. In chemistry, they talk about exergonic reactions, which is basically what this is. Don't worry about it. But if we have here reactants, and remember, these are the, it's also referred to as substrate. Reactants, substrate. I think the first slide I called this participants. It's going to go through a chemical reaction and become products. Now, some of this can be random, but if we have an enzyme in here, it's going to go faster. Okay? How does it go faster? Well, here's the reactants here at this level of potential energy. It's got to go through a chemical reaction to become products down here. The activation energy of a chemical reaction is the hill.
Okay.
The activation energy of a chemical reaction is the hill. It can't get down here until it gets up and over the hill. Activation energy. What enzymes do is they lower the activation energy. So here, do you see this kind of looks like what I have on the whiteboard? That hill is the activation energy. If you have an enzyme, the hill is shorter. And the reaction rates will naturally speed up because you don't have to put as much energy in it to make it go through. So without a catalyst or an enzyme, you got the blue hill. With the catalyst and the enzyme, you got the red hill. It's much a smaller hill to go over. The reaction rate goes faster. Any questions?
Even though the hill isn't as large, are you still getting the same amount of energy?
With less energy, the red reaction will be at the same speed as the blue reaction, which will require more energy to be at that speed.
Just because of an enzyme?
Right.
Okay.
So if the energy is equal,
Uh-huh.
and this is one of the reasons why, that's just part of it, it's important that our bodies are at 37 degrees centigrade. Is so that temperature has energy in it. Okay? So if you have the same 37 degrees centigrade, you have the same energy. Now if you shrink the hill, it's going to go faster.
Got it.
Okay? Lowers the size of the hump. Okay? Enzymes are extremely specific because they are proteins, and proteins are the huge boulders. And boulders have specific three-dimensional shapes. So if you look at this figure, you have the same substrate, reactant in the middle. 3 different enzymes are going to get you 3 different products because they're that specific, even if you start with the same substrate. If you've got different enzymes, you're going to get a different reaction with a different product. That's what this figure is telling you. And it's all because of lock and key. The very first slide last week. Wasn't we talking about specificity? It's just 3-dimensional shapes. You can't get a square peg in a round hole. Does that ring a bell?
Mm-hmm.
Look at this reaction here. Doesn't fit. There's no reaction. But if it does fit, it's going to have reaction. Look at this. Watch it. Is that anabolic or catabolic? These words mean something that you need to know for lecture exam 1. Is that reaction anabolic or catabolic? Catabolic. You have a big reactant and you got two small products. You tore it down. Does this make sense? You break it down, it's catabolism. Boom. One big reactant becomes two small products. Again, this is talking about that specific three-dimensional shape. And the three-dimensional shape is called the active site. That's where the reactant temporarily binds to the enzyme. And it fits like a lock and key. If it doesn't fit, there's no reaction. What kind of reaction is that in the animation? It is also catabolic. A big reactant is being split into two small products. It's breaking down. Some reactions require what's called a coenzyme. In this animation, it's the blue line. Okay, so if you read in the textbook about enzymes, they will bring up things like coenzymes. Right here, only this substrate C is going to fit. The other ones won't fit, no reaction. That's simple. All right. All proteins, all of them, including enzymes, are subject to denaturation. This is going to be relevant, not next week in lab, but the following week. In fact, next to the slide I would write lab 3. This is relevant for lab 3, which is a group lab report lab, so it'd be good to understand what denaturation is. Denaturation of a large protein is held together by various bonds, and we'll actually talk about that today. So when we talk about the chemistry of proteins, this is going to be relevant. But when you expose a protein to extremes in temperature or pH, or heavy metals, or high concentrations of chemicals, you're going to break some of those bonds that hold this thing into a specific three-dimensional shape. When that happens, the protein is permanently altered. If it is an enzyme, it is permanently disabled. Everybody got it? Permanently, like the best way to envision denaturation is cooking an egg. You're exposing the protein, most of it is albumin, to extreme temperatures. And then it goes from that snotty thing to something that's a little more solid, right? That's denatured egg. You made it hot, it cooked it, now it's denatured. If you put it in the fridge, does it go back to the snot?
No.
It is permanently altered. Everybody, that's the best way to understand the permanence of denaturation. Again, this will be covered in lab 3. This is big for group lab report. Most enzymes have these, for enzyme activity, close to a bell-shaped curve, where this is called the range of tolerance. In this case, it's from 0 to 60. If you're in between those temperatures, that enzyme will work. It works best usually close to the middle. But if you're outside, if you're above 60 or less than 0, it is denatured and it's permanently altered. Everybody got it? How to read that graph? Question?
Is it only enzymes that are permanently altered, or
No, all proteins.
all proteins?
Okay. But enzymes are included in that, so
Yeah.
they will do it too. And here's some of the bonds here in this picture. These are disulfide bonds, and they'll break. And that thing is never going to be the same shape. Everybody see that?
Mm-hmm.
We'll talk about some of those bonds that give proteins its shape later. All right, and then we got this concept of reaction kinetics. Go ahead and fill in the blanks and let me explain this. topic of chemical kinetics is this has to do with the chemical reaction speed and things that influence it. In a nutshell, that's the best, simplest way to explain it. This has to do with chemical reaction speeds and the things that can alter it. One of the things that alters it is substrate concentration. Remember, the bracket means concentration. So if you have higher substrate concentration, you're gonna get a higher reaction velocity up to a point. And that point is where the line goes horizontal. At that point, all the enzymes are working as fast as they can. You have reached substrate saturation. Everybody got it? It's not, the words are not on the PowerPoint, so you better be taking notes.
Is substrate saturation the where it peaks?
Where all the lines go horizontal. Just can't go above that. The enzymes are saturated. Doesn't matter how much higher, how much more substrate you have, the enzyme's the limiting factor. It's not gonna go any higher. It won't go higher unless you have more enzymes. Okay? Everybody understand the underlying terms here? I've covered the first two. We all good? Then you have enzyme activation and enzyme inhibition. There are things that will cause the enzyme to even speed up more than normal. That's the blue lines. And this is now steeper. However, you're just getting faster to saturation. Some of those things could be things like coenzymes. Some coenzymes will cause that black line to go blue. Everybody see that? There are other things that will compete for the active sites or block the active sites of the enzyme. And that will basically take out those enzymes. And not only are you going to get a more shallow rise, but in some cases where some active sites are now blocked, it'll never get up to the horizontal line. Because you have less functional enzymes. Everybody got it? Now if you physically block the active site, you take out that enzyme. Any questions?
Do we need to know what those inhibitors are, or just that there is certain things that inhibit enzymes?
Yes, the concept. You don't need to know specific activators or inhibitors. Okay. Go ahead and fill in the blanks.
Dehydration.
These are two examples of some of the reactions we're going to see in metabolism. They are completely the opposite. What do you do when you have paired opposite content?
You'll learn one.
Learn one, just learn the name of the other. So let's just learn dehydration. Basically, and its synonym is condensation reaction. Those are synonyms. Dehydration reaction, condensation reaction, they're both highlighted. You need to know both. And it's shown up here in the top figure. And like condensation, if you park your car outside, you wake up in the morning and it's covered in water. Condensation. It's like, the water came out of nowhere. No, the water was in the air. And when it cools, it'll actually come out of a gas and become liquid water. It settles on your car. But it is like water out of nowhere. Well, in this case, you got one reactant that has an H, you got the other reactant that has an OH, and they combine to become H2O. Water came out of nowhere. Does this make sense?
Mhm.
Kind of like condensation. Is this a catabolic, look at the picture, catabolic or anabolic reaction? It's anabolic. If you go from 3 and 1 to 4, that's building up. 2 smaller to 1 bigger is building up. 1 bigger to 2 smaller is breaking down. Everybody got that?
Mhm.
Whenever you do anabolism, you have to put energy in. And so you do have to put energy in this as well. Okay? And when you put a lot of energy, it says requires energy. That energy is stored in potential energy. These guys all have a lot of potential energy because you put a lot of energy into it to build them up. This is why when you eat, and everybody here, if you want to stay alive, you got to eat cells. If you don't eat cells, you're not alive. Okay, now if you're vegan or whatever, you can figure out how to do that. You're still killing cells. Nobody gets off this planet without killing cells. And that's because when you eat these and you break it down, you get energy, and then you're gonna make your own. But when you make your own, you gotta put energy into it and then store it. Everybody got that? Dehydration, condensation, it's anabolic. You gotta put energy in, it's required, and then you can store energy in large chains. The opposite is hydrolysis, in the bottom. That's all you should remember. It is exactly the opposite of the first one. You are inserting the water. The word hydrolysis means splitting by water. You're taking a larger reactant and you're splitting it into 2 smaller ones by inserting a water. H goes on one side, OH goes on the other side. It is exactly the opposite. When you do that, energy is released. Any questions? Am I going too fast? Is this boring?
No.
All right, let's go over the nutrients. Nutrients are also covered in your lecture packet. Starting on page 6. It also talks about anabolism, metabolism, enzymes, and cellular energy is ATP. This is the only energy your cells use. Well, not the only one, but it's the main one. Okay, the term carbohydrate means an H2O for every carbon. So that's the overall formula for carbohydrates. An H2O for every carbon. This is the primary energy source used by cells to make ATP. Again, inside the cell, it's only ATP. The links are monosaccharides. Most commonly, it's glucose and fructose, but there's a whole bunch. And they are C6H12O6, and H2O for every carbon. If you bind 2 links together, then you have disaccharides, 2 links. Table sugar is sucrose. It's a specific way of binding a glucose to a fructose. So it's a disaccharide. Your body will break down starch and turn it into maltose. And another disaccharide is lactose. Our digestive systems make an enzyme called lactase, which some people never make, and some people make a little, and then as they get older, they stop making it. And that's when people become lactose intolerant. Anybody heard that?
No.
Lactase is the enzyme. All right, let me just say one thing about sucrose, since I used to practice dentistry way back in the day. Oh, you know, and it's so funny because I hear what students say about me, which by the way, I don't give a shit about. But I hear, someone said that you got in trouble in dentistry and that's why you don't practice it. So, that's funny. I went on a medical mission to Central America, caught a parasite. 6 years later, I came down with rheumatoid arthritis and had to retire on disability. I was a good dentist. Some people say great, because in 12 years I owned my practice, I sold it for 5 times what I bought it for. So if anybody wants to say that I got in trouble in dentistry, you straighten them out. Well, because of what I know about microbiology and the bacteria that is commonly in supragingival plaque, when you expose them to any of these, yeah, the metabolic rates rise when you expose them to glucose, when you expose them to fructose, and lactose, and maltose. They like it, they go up. But when you expose them to sucrose, it skyrockets. Both their metabolic rate and the rate of mitosis. They go crazy. And the only reason why 85% of dentists in America have jobs is because of refined sucrose in our diets. It is a natural sugar, but it is rare on this planet if humans weren't involved. There's only a couple plants that make it, and the humans started to refine it. That's when we started to see dental problems. You dig up skulls from 1,000 years ago, these people didn't have floss, fluoride, toothbrushes, or dentists, and they didn't have cavities because they didn't have refined sucrose. A lot of the diabetes we have in Western culture is from sucrose. You take that thing away, you put it back in its natural concentrations in the real world, no problem. It is refining it that is the problem. The large chains are the carbohydrates out here. Now technically they're all carbohydrates. I tend to like to call carbohydrates the large chains. In this case, our cells make glycogen, plants make starch. Both are nothing but glucose links. But it's how you bind the glucose links together is the difference between glycogen and starch. Or cellulose, which, by the way, we don't make digestive enzymes for. You can't break down cellulose, which is why your mom told you to chew your corn, or you're gonna see it tomorrow. That corn husk is cellulose, and we can't break it down. These huge branch chains can be huge. Here we got this guy here. Each one of these little white things is a single ring glucose. This one right up here on the top is each one of these white guys. Or each one of these pink guys. Or each one of these black guys. You can see how huge these chains can get. But they're all glucose links. Everybody got carbohydrates? Single links on top, monosaccharides, disaccharides here. And then the large polysaccharide glycogen. But each one of these links is just a glucose ring. Right here, each one of these guys. Which one's bigger?
What are you asking between?
Glucose or glycogen?
Glucose.
Which one's bigger?
Glycogen.
Glycogen is the large branch chain. Glucose is a single link.
Okay.
Everybody got it?
I got it now.
Engage, people. Engage, because that's actually way simpler than the test questions I'll ask on lecture exam 1. Be engaged. Second is lipids. They are pretty diverse structurally. For you guys, there's one exception, but you can just think of lipids as being hydrophobic. Okay? There is one exception. The most common are triglycerides, which is why I say up here, this is the one example that doesn't really work well with chains and links. But a triglyceride has 3 links, 3 fatty acids, and they're bound together by the glycerol. and it is one of the more common lipids in your body, so it is the chief form of energy storage. This is the fat you think about when you think about getting fat. Lipids can also be structural. We've already had a lipid already this semester when we talked about the phospholipid. Remember how it has one head and two tails? So the phospholipid has two links. Okay? One's straight and one's bent. But this is the main thing in the phospholipid bilayer in that picture. Everybody got this? Triglyceride in the brown box, that's the glycerol. It's a 3-carbon molecule and then 3 fatty acids. Phospholipid, this is the phosphate group here. And this is what causes it to be polar. That is nitrogen ion. But you have negative and positive, it's polar. Got here in this case, a west pole different from an east pole. But that's why it's polar. Everybody got it? But the 2 long tails, and again, like in organic chemistry, they didn't write all the C's. These are all C's and H's going all the way out here. Okay? Another thing to talk about with fatty acids is this difference between saturated and unsaturated fatty acids. Didn't I write this on the whiteboard there in the first week? Now I put just a ... on the bottom. But the reason why it's called a fatty acid, you're gonna see this later, so I may as well show you now, is on one end of this thing, you have what's called a carboxyl group. And this carboxyl group tends to lose this H. When that H plus goes into the solution, it turns it acidic. Carboxyl group is acidic. So this is a fatty acid. Does that help make sense of the name now? Long hydrocarbon chain, and then boom, you got this acidic carboxyl group. Now, remember that carbon will always form 4 bonds. So if you look at what I drew on the whiteboard, there are 4 lines coming out of every C. Even down here, even this C. 1 here, and 2 to the oxygen. Carbon always forms 4 bonds. Because of that, just remember, only 4 lines can come out of every C. And because only 4 lines can come out of every C, if I have a double bond in the middle of the chain, I have to take 2 hydrogens off. Otherwise, there's going to be 5 lines out of those C's. Are you tracking me? If you take a double bond in here, you got to take 2 hydrogens off. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. If this is many carbons as you have, it can only fit 14 hydrogens max. For this length of a fatty acid. If it has 14 H's, it is saturated with hydrogen. Since this only has 12, this is an unsaturated fatty acid. Does that term now make sense? I'm explaining this as simply as you'll ever hear it. If you have more than one double bond, that is a polyunsaturated fatty acid. So you could write monounsaturated fatty acid there for the one. By the way, that's why one of the tails of the phospholipid is bent. It's bent because of a double bond. In a phospholipid, one of the fatty acid chains is saturated. One of the fatty acids right here is monounsaturated. You got one double bond. So that's why that one is bent. Here's the double bond right there. Saturated is straight, unsaturated is bent at the double bond. Did you guys notice that before?
Mm-mm.
Okay? So here's the difference between a monounsaturated fatty acid and a polyunsaturated fatty acid. This is relevant for cardiovascular disease. Everybody look at me. Dietary cholesterol is irrelevant. Okay? Doesn't matter if you eat cholesterol. What matters is, are the fats you eating saturated or unsaturated? And it's because what your liver will do with it. Okay? But dietary saturated fats have been linked to increased low density lipoproteins, LDLs. And that tends to form the atherosclerotic plaques of cardiovascular disease. Any questions? Again, the lipids are hydrophobic except for one example. Other types of lipids are ketones. Ketones are acidic, and diabetics tend to have high ketones. If you might actually hear the term diabetic ketoacidosis, because ketones are acidic. Write that next to that diamond. Ketones are acidic. Diabetic ketoacidosis. And in the textbook, there is a little box that will discuss that. Focus on diabetes. Steroids starts out as a long fatty acid. It gets a U-turn, goes through a couple other reactions. But it's like, here's that carboxyl group. So it's like this long fatty acid went all the way down here, made a U-turn, went around in here, then ended right there. Then enzymes cause there to be these cross covalent bonds. So you end up with 3 6-membered rings and 1 5. Do you see that in the picture? 3 6-membered rings, 1 5. It used to be one long fatty acid. And that's a steroid. But that's why steroids are hydrophobic. The one that is the exception is this, prostaglandins, or icosanoids. And then it's because it does a U-turn with a 5-member ring. That causes this molecule to actually have a north pole different from a south pole. Okay? Circle that word hydrophilic. This is the one exception. And lipids. This is the one exception that is not hydrophobic. Again, steroids, 6,6,6,5 used to be a fatty acid. But remember, we don't write all the C's in here. Every one of these corners is actually carbon. Everybody got it? Mm-hmm. And here's that prostaglandin that is actually polar. 3 is proteins. The links are amino acids. These are the structural components of the body. Whenever you see a body, you're looking mostly at proteins. And you do have to recognize visually for lecture exam 1, the NCC of an amino acid. Everybody got this? These are all the proteins that you see. Actin, myosin, is skeletal muscle. Collagen and keratin are all proteins. What's weird is that with keratin, that is the protein that's in your stratum corneum, what you see with your skin. It's the same protein of your nails. It's the same protein of your hair. All you do is you put those chains together differently, and that's the difference between nails, hair, and stratum corneum. And again, enzymes are all proteins. All right, here's the NCC you gotta recognize. You have the amino group on one side. Now, those molecules in that row there will form 3 bonds. Carbon will form 4. And then on the other end is that carboxyl group. And that's why this is the amino group here. Amino, and because this falls off, acid. That's why it's called amino acid. Okay? Now what separates the 20 amino acids in our body is the R group. So there's only 20 in our proteins. You don't need to worry about each amino acid structure. And there's 4 of them in this figure. You don't have to learn that. You do have to recognize the NCC. Do you see the NCC?
Mm-hmm.
Do you see the NCC on the whiteboard? Because you also have to understand how these links are bound together in a chain. Now, I'm not going to write all the lines here. You do a dehydration reaction or condensation reaction, and you pinch off some water. So you take off an OH and an H, and the water comes off. So we should put this out here. But each one of these guys here is referred to as a peptide bond. And you must be able to visually recognize peptide bonds. For lecture exam 1. It looks like that. Any questions? Okay, if you have the sheet that has the essays, you should pull that out. Got it on Monday. And you want to look at the very first one, number 1. The 4 levels of protein structure. It is copied and pasted from this slide. Primary protein structure, that's the 1 degree. Primary protein structure is for my peptide bonds. It's the specific order of the amino acids. Now this is referred to as a small polypeptide with 4 links. But if you notice, whenever you get a protein, you always have an amino end and a carboxyl group end. and it could be 300 links long, it'll still have an amino end and a carboxyl end. Can you just imagine me drawing this all the way out the door? This would still be the amino end. Everybody got that? Everybody got that?
Yes.
Engage. Again, if you guys want to go into programs, these people are not going to wait for you. You got to keep up. Okay, so the specific amino acid sequence formed by the peptide bonds in purple, that is primary protein structure. As this chain gets longer and longer, these guys, these oxygens are going to like the hydrogens, because these are negative, remember water, and these are positive, they're just protons. Everybody got it? So as it gets longer and longer, you're going to see this attraction between negative oxygens and positive hydrogens. We call those hydrogen bonds. They are weak. But if you form enough of them, you can get either a helical structure, we call it alpha helix, or this thing that looks like you folded a piece of paper over and over again called a beta pleated sheet. Okay? This one is only showing you an alpha helix. But that is secondary structure, secondary protein structure. If the protein gets really long, so not all proteins have this, but some do. If it gets really long, there'll be other bonds, hydrogen bonds, ionic bonds, those disulfide bonds we saw in the denaturation figure, as well as van der Waals forces. You guys know what those are? You need for me to explain it to you? Remember that certain things are hydrophilic, they love water. Certain things that are not polar are hydrophobic. Van der Waals forces is when you got a real long chain. If you have something that's hydrophilic, and you have another area of the chain that's hydrophobic, it will move away from this part. Or if you have another part that is hydrophilic, it'll be attracted to this other part that's hydrophilic, because water-loving things love each other. Everybody got it? So it's an attraction of hydrophilic to hydrophilic, or an attraction to hydrophobic, or repulsion between hydrophilic and hydrophobic. That is van der Waals forces. As simply as I can state it. Okay? Well, anyway, this really long chain going out the door will now have sections that are alpha helix and beta pleated sheets. It bends back and forth and back and forth and back and forth until it becomes a specific glob shape. Glob is a good thing to remember. In fact, next to tertiary structure, the third diamond, write glob. It's a specific three-dimensional glob shape, from whence we get the term like hemoglobin. But the thing about this here is that this is a single chain. First, second, and tertiary structures are all single chains. Only a few in our bodies will match up multiple chains in a specific arrangement, and that is quaternary protein structure. And two examples of that is hemoglobin, that has 4 globs, and insulin, that has 2. But you have multiple chains. Is that not what is on your essays? Now, this handout is giving you the content you need for essays. On the blue book, you must write it out in complete sentences. Everybody look at me. Everybody look at me. For lecture exam 1, this is just the content. You must write them in complete sentences with this content. Are we clear? All right, so now on the slide, put a little arrow off to the side from the second and third one, and write denaturation. Denaturation. Lines going to the second and third one, because denaturation breaks weak hydrogen bonds. And where are hydrogen bonds? Second protein structure and tertiary protein structure. That's why those are the two that get broken up with denaturation, and that's why that glob now has a different shape. Does that help to tie to denaturation?
Mm-hmm.
Any questions? There's primary up on top. There's secondary on the bottom, alpha helix. This is what a beta pleated sheet looks like. It's like you took a piece of paper and bent it, folded it like a fan. Isn't that what that kind of looks like? Tertiary is a specific glob shape that has sections of pleated sheet and alpha helix. And quaternary, you have to have more than one protein chain. All the way up to tertiary protein structure, it's just a single chain. Once you get to quaternary, you must have more than one. Hemoglobin has 4, insulin has 2, so they both qualify as quaternary protein structures. Any questions? Okay. And this is a little bit more on denaturation. Again, you can have other things besides extremes and pH and temperature. For lab 3, we're only going to work with extremes in temperature and pH. We're not going to work with heavy metals or reducing agents or alcohol and things that could hurt you. I guess if you stick your hand in boiling water, that would hurt. Okay? But remember, now on the previous slide, did you put a line to the primary protein structure? Did you put a line from the primary protein structures of denaturation? No. Only second and tertiary gets altered. Primary protein structure is intact. The order of amino acids are still the same. But it says right there, alters the three-dimensional shape of the protein without altering primary protein structure. It's still the same. And it's good. A good illustration is frying an egg. Again, this is permanent. Even if you put it back in the fridge, it's not going to go back to that snot. Okay? All right. We're doing good. Any questions on the what we covered so far with cell chemistry? Okay, I'm going to set up the first, everything up until now has been preliminary stuff. Are there any questions on the preliminary stuff? Because now we are going to go into the first of 2 major topics for lecture exam 1. Metabolism, or cellular respiration, and DNA. In fact, you might want to write this down now. First big topic is metabolism. Second big topic is DNA. Under DNA are 2 subtopics: protein synthesis and DNA and cell replication. The bottom line that I'm going to do for you with metabolism is I'm going to start here with monosaccharides and how our cells use monosaccharides to generate 36 ATP. I'm going to build that framework first, which is going to require 3 major steps. But it's how our cells will take glucose, which is a monosaccharide, and make 36 ATP out of it as an ideal yield. Once I establish that framework, and this will be on Monday, then I'll plug in the other two. You are not responsible for how we make ATP out of nucleic acids and nucleotides. Our cells can, but I'm just gonna concentrate on the nutrients, 1, 2, and 3, and it's these guys. Are we clear? Okay. This is the overall formula. Remember, glucose has 6 carbons, and there's a H2O for every carbon. You're gonna need oxygen to completely oxidize the glucose, and your cells are gonna convert that to 6 carbon dioxides and 6 waters. But by doing that, it's also gonna put 36 inorganic phosphates, that's the I, no carbon here, on the 36 ADPs to generate 36 ATPs. Now, 36 is an ideal yield. No cell actually does that, but that's what we're gonna go with. Also, by the way, I gotta be quicker about this. This is why this is called cellular respiration, because the cells are gotta utilize oxygen and it's gonna generate carbon dioxide. And if all your trillions of cells are doing that, that's why you have to take in oxygen, and when you breathe out, why, there's more CO2. Does this make sense? Everybody look at me. This is not respiration. Yeah, well, if you take a life-saving course, they might say that's respiration. Not in medicine. This is ventilation. The gas exchange at the alveolar capillaries is external respiration. The gas exchange in systemic capillaries is internal respiration. But what we are going to be talking about is what's happening inside the cell, and it's this. And that's cellular respiration. Okay, everybody got these terms? Well, when we do this, we're going to cover some preliminary principles. I'm going to cover some today before we end. And then we're going to cover the 3 stages of cellular respiration. We'll start with glucose. And that is what we're going to do on Monday. But here it is, the overall thing. Starting with glucose, which is a 6-membered ring. We're going to need oxygen, we're going to get out CO2 and water, and about 36 ATP. If you want to get the 3 stages now, it's on this figure. Glycolysis is first. Krebs cycle is second. The ETS, electron transport system, is third, which, by the way, is where the oxygens get burned. Okay, and you're gonna get 36 ATP, but look at where the payoff is. It's actually in the 3rd stage. You only get 2 in the first, 2 in the second, 32 in the 3rd. That's the payoff. Does everybody see that in the figure? This is from your textbook, the overall formula. Preliminary principles. Go ahead and fill in the blanks. First is a transfer of high energy electrons, but it's not free electrons. They're in the form of hydrogen, because hydrogen is only a proton and an electron. So it's in hydrogen, proton and electron. That's what it says here. It's often in the form of hydrogen ions, proton and electron. But that electron is the high energy electron. Now, some of these terms might be confusing. You gotta find a way of making it make sense so you don't mix it up. If you gain electrons, that molecule that gains electrons is reduced. If you lose the electron, that molecule is oxidized, and they always happen together. There's never one without the other. Well, that's why we call these redox reactions. Oxidation reduction reactions always go together because it's transferring that high energy electron from one to the other. Goes from molecule A to molecule B. B that gained the electrons are reduced. A that lost the electrons are oxidized. And the easiest way to remember it is the mnemonic Leo-Gur, as like Leo the Lion says, Gur. Lose electron, oxidize. Gain electron, reduced. And you might think, if you gain something, how do you get reduced? Well, maybe just think about the fact that electrons are negative. You've gained and negative, you're reduced. Does that help?
Mhm.
Again, you never have one without the other. Those electrons have gotta go somewhere. So here we got this being oxidized and this being reduced. Always together. In our cells, this is going to be accomplished by dinucleotides. I think these are also ribose as well. Yeah. So these are, ATP is a single, but these are double. These are also RNA nucleotides, but they're double nucleotides. And each can take a pair of hydrogens, a pair of electrons. This is where NAD picks it up, right here, there's two of them. This is where FAD picks it up, 1, 2. Now, there are multiple ways that textbooks and online sites will designate these. For the lecture exam, it's not gonna be a big deal. Just know that if you have the H's, you have the electrons. If you have the H's, you have the electrons. If you gain the electrons, you are high energy, because these are high energy electrons. This is the high energy reduced form. Because it gained the electrons. It'll be really helpful, especially when we get to that third stage, the electron transport chain. You have this straight, right here. Because they can exist in both forms. Bottom bullet, they can either be reducing or oxidizing agents. Right here. Some reactions we're going to see in the 3 stages. One is a decarboxylation reaction. And that's removing a carbon in the form of CO2. That's what you're going to be breathing out. Because this is anabolic or catabolic decarboxylation. Look at this figure up here. How many carbons here as a reactant? 3. How many carbons here as a product? 2, with the third one going off here. A larger reactant got split into 2 smaller products. 3 went to 2 and 1. What kind of reaction is that? Catabolic, so energy is released, energy out. But this getting 36 ADPs to become 36 ATPs is 36 phosphorylation reactions. You're adding a high energy phosphate group onto ADP. Substrates. Substrates. Here we're talking about carbons. If you make ATPs where there are carbons, that is substrate level phosphorylation. So if you've got carbons, if you're making ATP there, that's substrate level phosphorylation. That's highlighted. If there are no carbons there, and you make ATP in this 3 stages, that's oxidative phosphorylation. Okay, let's go back to this one. There are carbons here, that's substrate level phosphorylation. There are carbons here, substrate level phosphorylation, but by the time you're done with the Krebs cycle, all 6 carbons here in glucose have now become 6 CO2s. So there's no carbons left here. This 32 is oxidative phosphorylation. You should write that down. Substrate level phosphorylation. This is substrate level phosphorylation, the 2 and the 2. But the 32 is oxidative phosphorylation. The carbons are all gone. In fact, if you look at your textbook, they don't even call this the electron transport system. They actually call it oxidative phosphorylation. Everybody got it now? Mm-hmm. Just compare this figure from this slide, very bottom, to this figure at the very bottom. It is oxidative phosphorylation. But these two and these two are substrate level phosphorylation. Everybody got it? Because once you're done with the Krebs cycle, you are done with the carbons. But you started out with 6, 2 go out here, 4 go out here. There's your 6. Everybody see that? Mm-hmm. Okay, we're going to start with glucose. I'm going to build that framework first. And we have 3 sources of glucose. Facilitated diffusion through channels. Breaking off glucose links off of glycogen. Glycogenolysis, you're gonna have to keep these glue and gly words straight. There's a lot of them. But glycogen is the branch chain, and if you split it up, it's glycogenolysis. That's the second source. Glycogenolysis, breaking off glucose links off the glycogen. And then there's gluconeogenesis. Let's split up this word so it makes sense. Gluco is glucose. Neo means new. Genesis means creation out of nowhere. Right? Isn't that a biblical term, Genesis? Mm-hmm. What that means for this is that you are making new glucose from non-carbohydrate sources. You're making new glucose from non-carbohydrate sources, which is why gluconeogenesis doesn't count, since glycogen is a carbohydrate. That doesn't count for gluconeogenesis because glycogen is a carbohydrate. This is you're making new glucose from things that are not carbohydrates, and your liver can do that. Everybody got gluconeogenesis? Okay, so here's the 3 stages in order: glycolysis, Krebs cycle, electron transport system. We're gonna go in more detail. Believe you me, because I've taken biochemistry, we're only scratching the surface. Okay. Everybody got the 3 stages? Those are the 3 stages right here. Glycolysis, Krebs cycle, it does have a preceding blinking step, and then electron transport system. And you might want to check, and if any of these links don't work, let me know.